This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 171, recorded on February 15th, 2018. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm well. How are you doing?
Okay. Hanging in there.
Everything good in California?
Yes, sir.
The world
Not
is
bad.
all good. Very good. Here in New York, it's winter.
What's that?
Yeah, it's what you used to know that in Boston, right?
That's right. I forgot.
Also joining us from Charleston, South Carolina, Michael Schmidt,
Hello, everyone.
where it's probably California-like weather.
It's beginning to, the trees are in bloom.
Are you serious? In February?
When the Japanese magnolias begin to show their buds, it's either a sure sign that there's a frost next week.
Wow.
I mean, it's the flowering trees are flowering. It's quite spectacular. The crab apples have blossoms on it.
Nice.
It's pretty.
Very good. I hope maybe one day I'll get down there to visit.
Maybe.
Would be nice. And that is our crew for today. Michelle is busy doing important things, but we will shoulder on and tell you some cool microbiology. We have two very good stories, as always. You know, when you do 4 stories a month, you can't help but have 4 good stories because so much microbiology is published, right, that we can figure out at least 4 good stories. We could even do more. But our first snippet comes to us from Hannah, who writes, "Dear TWiM hosts, this new Nature paper about stingless bees demonstrates that they need a steroid from a mutualistic brood cell fungus to pupate. I thought it might make a good snippet." And Hannah was indeed right. It will make a great snippet. It's a really cool paper. And I think this is the kind of story that Elio loves.
I sure do. I love tales of symbiosis. Somehow, it seems to me that tickles the mind more than a single genome at one at a time. When you put 2 genomes together, 2 organisms together, they interact. It becomes interesting no matter what they do.
Well, it's how the world works. Always better when people collaborate, right? And
Yeah,
when instead of doing it, you can do it yourself, but wow, it's so much better when you collaborate. So this is published in Scientific Reports, which is an open access publication of Nature, so you can find this online. And the first author is Camila Paludo, and the 2 last authors are John Clardy and Monica Pupo. And Roberto Coulter is on this paper, Elio.
Yeah, and so is Cameron Curry. Big guy on the fungus-ant relationships.
That's right. So this comes out of the University of São Paulo, the Brazilian Agricultural Research Corporation, Harvard Medical School, the University of Wisconsin. So it is a multi-university and country work. And this is about stingless bees. Now, let me tell you a little bit about bees, because this is very interesting. But bees are believed to have originated about 110 to 130 million years ago. And that's about the time when angiosperms became the dominant flowering plants. And there, all of a sudden, there was a lot to eat. So the ancestors of bees, instead of being predators, they became eaters of plants, phytophagous. So all of a sudden, you have all these flowering plants, and now you have an emergence of bees from wasps, and the bees diversify. Did you know that there are 25,000 known species of bees on the planet?
I thought there were only honeybees. That I'll get a letter for that.
No, there are lots of bees. There's some that don't sting, in fact, which is the one we're going to talk about today. Now, as everyone probably knows, bees consume pollen and honey, but recently a Brazilian bee was found that also needs to eat fungal cells in order to survive. This is called the stingless bee, Scaptotrigona depilis. It has to eat a specific brood fungus during its larval stage. When the eggs of this bee hatch, you can observe a white microbial growth between the cell wall and the surface of the food supply. And people thought, this is a pathogenic fungus, must be bad. But it turned out that it's a symbiotic fungus that's eaten by the larvae and is needed to complete development. And subsequently, it was similar idea. A fungus that is needed for development was found in other stingless bees as well. Now, I didn't even know there were stingless bees. So this is all new to me. So what is a fungus doing? Well, symbioses are multifactorial. They can provide nutrition. They can supply defenses. They can communicate. And there's some other examples of fungal symbioses. The famous one is atine ants, which cultivate fungi for food. They have fungal gardens, and these gardens are protected from other parasitic fungi invading them by actinobacteria that produce small molecules that inhibit those other fungi, but not the fungi that the ants need. So it can be really interesting, complicated arrangements here in the world. So what do you need the fungus for? Well, in order to to molt, the bees required ecdysteroids, which are steroid-derived hormones, and those induce the transformations you need to go from an immature individual, all the way to an adult. Insects can't synthesize sterols, so they have to get it in their diet. They have to make ecdysteroids from dietary sterols. And that's what this paper is about. If you want the bottom line, they find that a certain species of fungus, Zygosaccharomyces, it's the fungus that is eaten by these stingless bees, and that provides ergosterol to the bees so that they can make sterols, that they can make ecdysterols to aid in their development.
It's interesting that they had historically there was a wrong turn, because people thought that the fungus called Monascus.
That's right.
Does the job. In reality, what they thought it was a pathogen, I guess. But anyhow, they found it.
Yeah, that's right. That's absolutely right. They thought it was Monascus. So what they do here is they have they collect this fungal growth that is accumulating inside the brood cells, and this is what the larvae are going to eat. They collect it. They tried to grow it, and it wouldn't grow, but they thought maybe the brood cells are filled with high osmolarity food supply. So they said, let's use high osmolarity medium. And
makes sense.
They use 30% glucose. That's a lot, right?
That's a lot.
That's syrupy.
Syrupy. And it grows. And they can isolate it from the food, and they can sequence it. They can sequence the 18S and 26S RNA, and turns out to be Zygosaccharomyces. And it's not, as Elio said before, Monascus. So they can find some,
which is another yeast.
It's another yeast.
Yes.
And in fact, here they looked for Monascus. They couldn't find it. They just find Zygosaccharomyces. So Zygosaccharomyces is the one that's eaten by the larvae of these bees. Very interesting. And they have some nice pictures of the mycelium of this fungus.
Well, it grows in very sort of unusual ways, doesn't it?
Yeah. You can tell. You probably can tell us better than I can. Why is it unusual?
Well, it makes a pseudomycelium, which is not known to happen in Zygosaccharomyces.
Yeah.
Saccharomyces doesn't typically make a mycelium or a pseudomycelium, which is a short mycelium. And I don't know, Zygosaccharomyces was also not known not to do it. So this is, they say it's unusual.
Yeah. Normally it would be dividing its cells, right?
Yeah.
So the first question they ask is, can we feed Zygosaccharomyces to the larvae, and will it substitute for whatever happens in nature, the real thing? So they grow up. They actually collect larvae and make sure there's no fungus associated with them. So they get eggs and they move them out of where they were laid so that there's no fungus associated with them. And they show that there's no fungus there. and then they allow the eggs to hatch, and so they have larvae, and then they give them Zygosaccharomyces. They inoculate them with
1 microliter.
It's 1 microliter. I guess they are injecting it into the larvae, and this is grown up, of course, in the lab.
They just added to the 96 wells.
That's right. They put it right in the well, yeah. Along with the larvae, and then they say what percentage of larvae complete metamorphosis. So they get 71% with their Zygosaccharomyces, and it's 6% if you don't inoculate them with Zygosaccharomyces. And then those 6%, eventually they die anyway. They don't live. And the others, of course, go on and develop into bees. So this Zygosaccharomyces gives them what they need to mature, to go through their.
It's really pretty remarkable. It's a remarkable result, isn't it?
Yeah, I think it's very cool. And in fact, when they got this, I bet they were really excited, right?
They better be. I mean, who thought of this?
No, they were surprised. I mean, this is all new, right? So they were pretty surprised. So then they say, what is in the Zygosaccharomyces? So they say, it must be sterols of some kind, because the larvae will take the sterols and make ecdysteroids, which are needed for molting. So they extract sterols from fungus and they analyze it by mass spectrometry, which nowadays is pretty easy to do. And they find the major sterol is ergosterol. So then they take pure ergosterol and they add it to larvae, and they get at 2.5 micromolar, 65% pupation. And that's compared to 71% where they eat fungus. So it's not bad. And it's 8% when you don't give anything to the larvae, as you heard before. So ergosterol or ergosterol, I guess you could say it. What's the right way to say it, Michael?
Ergosterol.
Ergosterol. It supports larval development. So presumably they eat the ergosterol, which is produced by the fungus, and they convert it to ecdysones, which are then needed for the molting stages. And in fact, they find some of these in pupae collected from different colonies. They can find various sterols in them by mass spectrometry. And that's it, really. And so you need this bee, and probably a lot of other bees out there in the wild need fungus to undergo development. This is how they've evolved over the millennia to need this. And what I found the most compelling is that they say, farmers growing crops, they use antifungal pesticides.
Lots of them.
And what is that doing to these poor bees, right?
And do we know if that could be part of the reason for colony collapse of, just having fungicides out in the wild, and you may be selecting for a fungus that no longer makes these important steroids that, other types of bees may or may not need.
Yeah.
And, it's a continuum.
They say here that we should test the safety of antifungals mainly for wild bees, because I don't know if the honeybees need a fungus. Who knows, right? Maybe they could.
I mean,
I don't know.
I think, the microbiome, this is another example of.
This is a mycobiome, right?
A MYCO, a mycobiome. Well, it is.
Yeah.
And it really illustrates the, 2 + 2 = 12. And, you really see the codependence of higher organisms that do more complicated things like harvesting nectar and converting it to honey and fertilizing our plants. It really illustrates the importance of one member of a particular chain.
Yeah.
Everything is interlinked. And this, yes, it's a great story. Thank you, Hannah, for bringing it to our attention. It is a perfect snippet. And I'm sure we'll hear more about this story. Elio, you haven't talked to Roberto at all about this, have you?
No, and he's an author in this paper, which is totally surprising.
Yeah.
But Roberto has a way of researching every corner of the microbial world.
That's nice. Well, if you can get support for it, that's great.
That's right.
All right. Now, Michael, actually, Michael also found this paper. So at the same time you had sent it to me, Michael, Hannah did. And then you also have the other one, which I said, why don't you do that one, Michael?
Yeah, and the one I'm picking up is I traveled last week, so I had the occasion to be contained in an aluminum tube for 2 hours, and so I could read uninterrupted without email or anything else. And
you can get Wi-Fi on the plane.
I make sure it's off. I make sure it's off. It's the only peace and quiet you get these days. So this paper is from Science, and it was in the second of February issue, and it's entitled "Patients with Familial Edematous Polypopsis Harbor Colonic Biofilms Containing Tumorigenic Bacteria." And it's from a group at Johns Hopkins University in Baltimore. The first author is Christine DeHaa, and we'll have more to say about Dr. DeHaa, if I'm not masticating her name. Yeah. And she's in a large group led by Cynthia Sears. And it's a really fascinating story or study that shows the promise of translational science. It's a story of colon cancer, which unfortunately is the second leading cause of cancer death in the United States. The last time they rolled up the statistics, the number of deaths per 100,000 for colorectal cancer or colon cancer was about 15 per 100,000 men and women. And the percentage of colorectal cancer deaths is highest amongst people aged 75 to 84. And, thinking about the background of the story and why it matters and why I gave you the age statistics will become apparent as we tease the paper apart. But on average, the lifetime risk of developing colon cancer is about 1 in 23 for men and women. So it's at the same rate that we get healthcare-acquired infections, about 4.5%. So this is a pretty big deal. So the cancer rate, however, unlike healthcare-acquired infection, varies widely according to individual risk factors. And today we're going to talk about the role that microbes play in this process in a genetically predisposed cohort or population of patients. And as we know, the colon contains trillions of bacteria that are isolated from the colonic epithelial layer where the colon cancer will develop by this dense layer of mucus, and that there are bacteria out there like Akkermansia mucinophilia that we talked about on TWiM 110. And we entitled that episode "Exploring Unseen Life with Unpronounceable Words." And
that was Elio's. That was Elio's.
And that use mucin as the sole source of carbon and nitrogen, and use gastric mucin to colonize the GI tracts of a number of animals. So we know about bacteria that can eat mucin, but these trillions of bacteria that are in our colon can produce toxins, and some of those toxins are mutagenic and or carcinogenic. And this magic mucus layer between the colon's lumen and the epithelial layer where the cancer would develop promotes tolerance to foreign antigens by limiting this bacterial epithelial cell contact. Mm-hmm. And that's the gist of this story is understanding this dynamic of how do bacteria that make a toxin that can cause a mutagenic event then develop into cancer. And, of course, there's inflammation involved, and the colon is a nice mucosal surface. And so you have mucosal inflammatory responses coming in. So you begin to see it becomes a complicated story. But this group has taken it apart nicely for us in this genetically limited group because, we have the genes of the host, namely humans, interacting. And so I liken the mucus layer to the shields that we are always hearing about in every space opera we have ever seen or heard, whether it be Star Wars, Star Trek, or even the newest crazy space opera out there, Orville, by, Family Guy's fame, Seth MacFarlane. And so there's always a need for a shield to protect the human inhabitants of the ship in the case of the space operas, or in the case of us, where our colon is no different than a spaceship, and this mucus layer is the answer or the shield. And so the authors tell us that we know that when bacteria breach this colonic mucus layer, with in some cases biofilm formation, this then fosters chronic mucosal inflammation, which is bad. And in fact, this group has previously shown that biofilms on normal mucosa of sporadic colorectal cancer patients were associated with this if you will, oncogenic state, suggesting that biofilm formation at this important epithelial layer is an important event in influencing the development of colorectal cancer. To summarize the background, colorectal cancer, individuals with sporadic, so they're not genetically predisposed for it, frequently harbor abnormalities in the composition of their gut, and inflammation is often associated with it. And the microbes making the toxins get past the shield, and they result in the development of the cancer. So now here's where we get to today's story, and it's broken down into 3 parts. The first is...
Before you start, I don't want to steal your thunder because you're going to probably get to say this, but in a way, this is a remarkable paper because it is a very thorough examination that leads to really solid conclusions in a field that didn't always have that characteristic.
And that's why I picked it, because of precisely why Elio brings this up. It's because they lay this out. They start with human observations. They go to not 1, but 2 mouse models. Then they take it to tissue culture. And then they spin it out at the end, putting it all together for us in a straightforward model. And so that's why I started out with the Star Wars metaphor, talking about the mucus layer. And the mucus layer is the key. So we're going to start with the observations that they give us in the beginning, because it really sets the tone. So the observations of the human condition, they're going to test a hypothesis, and they really get to see what happens. To start, the microbiota associated with precancer lesions in hereditary colorectal cancer remains largely unknown. It only accounts for about 5% of the, I'm going to call colorectal cancer from now on, CRC. So 5%, or about 1 in 20 of the CRC patients. And of these hereditary cancers, colon cancers, the familial edematous polyposis, or FAP, only accounts for a subset of that 5%, or it again is about 5%. So overall, if you have 400 colorectal cancers, one of them will likely be an FAP causing cancer. So it's not a really big cohort, but this is where this paper excels, because they have access to these samples. And it doesn't diminish the wonder and significance of the observations and the model that the authors are laying out for us. So now recall that biofilm formation is an important epithelial event influencing CRC. So here, to test the hypothesis that the biofilm formation may be an early event in the progression of hereditary colon cancer, they examined the mucosa of these FAP patients after a clinically indicated colectomy. So they have tissue samples, and they're going to simply ask the question, who's there? And so through the usual methods that we have talked a lot about on TWiM, they're using 16S ribosomal RNA gene probes. They're adding a fluorescence tag to it, so it will light up under the microscope. And they start out with a probe that will light up many things, or a pan-bacterial 16S ribosomal RNA fluorescence probe using this in situ hybridization. So each FAP patient exhibited bacterial invasion through the mucus layer scattered along this colonic acid.
That's not right. That's not normal, right?
No, that's abnormal, and that's indicative that the shields have failed.
Yep.
And so why have the shields failed? And I should point out that Michelle,
causative or consequence, right?
it's the chicken or the egg. We don't know which yet, but they're going to tell us. They're actually going to tell us it was actually the chicken that is doing this. And it's really fascinating how they get us there. And I should point out that Michelle was very intrigued by this. She contacted the first author, and Dr. Duhay is an expert in imaging and fish models, and she's actually patented the process of examining these tissue samples on the direct biofilm detection of tissue sections.
You can imagine that would be a clinical assay that would be used to look at risk for cancer, right? Yeah,
absolutely. Because every time those of us over 50 years of age have experienced the hose,
none of us three, of course.
Oh no, I've actually experienced.
I'm just kidding.
I'll freely admit that
I've had four. I've had four.
And so, if you have a polyp, the beauty of the colonoscopy is they can take the polyp off and examine it. So this is where this technology can come into play. Now, here's our first curious observation. Unlike the continuous mucosal biofilms in sporadic CRC patients, so in a CRC patient that's not genetically predisposed, they have this continuous biofilm that's associated with the colorectal cancer. So it goes along and it's just not isolated. The tissues, however, from the FAP patients displayed patchy bacterial mucus invasion, and the average length was only 150 micrometers on 70% of the surgically resected colon specimens collected from 4 of 6 hereditary tumor patients.
Hey, Michael, how do they know that cutting these out of the patients didn't cause this?
I don't know. That's for the colorectal guys to talk about.
Do they have some normal?
They snip it very fast.
Oh no, they control it.
They control it. They have normal tissue.
Yeah, this is a well-controlled study. They have if not more normal controls to go along with this.
All right.
The other substantial observation of this FAP cohort was that one of the FAP patients who received a course of prophylactic antibiotics the night before surgery, when they looked at his or hers, the biofilm was not detected in this mucus layer. So the antibiotics effectively, quote unquote, presumably could have cured this, but they don't touch on that. So that I thought was really interesting that, a course of antibiotics could eliminate it, but I don't think that's necessarily the case because biofilms are typically refractory to antibiotics. So I think that's just a curious observation. But as you can quite imagine, they next asked, what microbes were present in this novel mucus penetrating biofilm? So again, instead of using a pan-bacterial 16S probe, they now use probes that are designed to recognize the major phyla associated with biofilms of the sporadic CRC, the quote normal cancer, if you will. And the usual suspects they have the probes for Bacteroides and Prevotella, the Proteobacter, the Lactospiraceae, and Fusobacteria. And notably, the FAP biofilms of these genetically predisposed individuals for colorectal cancer were composed predominantly of mucus-invasive Proteobacter. And those are things like E. coli and Salmonella and Vibrio. And they were responsible for about 60 to 70% of the community associated with these predominantly mucus-invasive biofilms. And then the other 10 to 32% were Bacteroides, which is very unusual. And the normal subjects that we consider to be causing problems like Fusobacteria were not detected. But
this is really a novel finding, isn't it?
Yes,
Michael. This was not suspected before.
wait till you hear who the players are. And I'm just going to jump to the players. The player was E. coli.
Amazing.
E. coli.
Yep.
E. coli was the proteobacterium that was doing this. Good old roly-poly coli.
And also in feces, it's a minor component.
Absolutely.
There's very little E. coli.
This was what so remarkable associated with the stories. Now,
Right.
One of their most disturbing results was they saw invasion of this epithelial cell layer by biofilm community members was detected in all patients harboring the biofilms, which was similar to what was seen in these sporadic CRC patients. So now we have the human observations from the FAP patients. So summarizing this, screening FAP patients, they learned that E. coli and
at this point, remind everybody what FAP stands for. You said it earlier, but
it stands for familial
adenomatous polyposis.
That's it. And that's why they call it FAP. And it's basically adenocarcinoma. So E. coli and Bacteroides have penetrated the mucus layer in these bugs. So they next ask, what's up? Well, there's strong experimental evidence out in the literature supporting the carcinogenic potential of molecular subtypes of both E. coli and B. frag. And we know that E. coli containing one of these genotoxic islands, and it was the one that contained a polyketide synthase. So in this genomic island, there's a gene that encodes the synthesis for colibactin, which is a genotoxin. So this induces DNA damage in vitro and in vivo, along with tumorigenesis in an azoxymethane-treated interleukin-10 deficient mouse. To suffice to say, E. coli makes a toxin that can cause cancer. The other thing is there was an enterotoxic B. frag, which they abbreviate ETBF, that also induces colon tumorigenesis in another mouse model. So I know you can see the next set of experiments coming up because they're going to take us to these mouse models asking questions. But before we get there, there's still some human results that the authors think we need to learn about before we can go to the animal model. And again, this illustrates how carefully they assemble this story. So human epidemiological studies have associated both the B. frag, that is this enterotoxigenic variant, and this polyketide synthesizing island E. coli with inflammatory bowel disease and then sporadic colorectal cancer. So they have this frozen bank of mucosal tissues from these 25 patients. And so they simply ask the question, can they culture out of this frozen bank of mucosal tissue the E. coli and the B. frag that have these toxins? Well, as you might guess, since they're telling us about this, the mucosa of the FAP patients was significantly associated with the genotoxic producing E. coli to the tune of 68%. And the toxin producing Bacteroides was present at 60% to compare to the mucosa of healthy subjects. And there was no preferential association of the enterotoxic B. frag or the PKS positive E. coli with polyp or normal mucosa from FAP patients, and that this mucosal association occurred at a higher rate than what was expected to occur randomly. So they're building on this.
This is really big news.
Oh, yeah. This is the microbiome's potential realized.
Right. Adverse potential, right?
I'll get letters for that too. So then they
this is like finding a new pathogen. It's like finding a new pathogen.
It's defining a new pathogenic potential for E. coli that we would normally think about. So the authors then transition from their human observational studies to their animal models.
Hey, Michael, can I ask you a question? So this E. coli with the toxin, right? Do you and I have this? What's the frequency in the population?
I don't know.
I wonder. So, I mean, it's, as we'll see, it's a problem to have this, but
yes,
I just wonder
that's how they end the paper.
Yeah.
That's how they end the paper.
Okay.
But I don't know what the frequency is, and that would be very good to effectively look at. The other issue is, since this is on one of these, if you will, pathogenicity islands, what's the selective pressure for E. coli to maintain this extra luggage in its genome in this particular niche of the large intestine? And so they didn't get into that aspect as well, but that's for the molecular ecologists to begin to debate. So they now next take us into their beautiful mouse models. And the first is a well-established model for cancer in mice. It's azoxymethane without dextran sulfate. And then the other model is the APC, which stands for edematous. It's the same gene.
Polyposis coli.
That's it. Gets mutated. I can't keep all the cancer genes straight in my head. And so they then get to that. And that's also got a deletion to test the hypothesis that the PKS genome-containing E. coli that makes this genotoxic product, as long as the enterotoxic B. frag, co-colonization, they say they have to get together, enhances colon tumorigenesis. So they do the mix and match experiments. They have E. coli by itself. They have the enterotoxic B. frag by itself, and they take us through that. And
they're just feeding this to the mice, right?
That's right. And they point out that the spontaneous rate of tumor formation using either model is very low, so that's good. So it's just garden variety SPF specific pathogen-free wild-type mice were treated with the cancer-inducing compounds azoxymethane, and then they had monoinoculated or co-inoculated with these particular strains. They first report that the colonization was similar under mono or co-colonization, and the bacteria remained in the colon until tumor formation was assessed at 15 weeks. So you can put a lot of bacteria into model systems, and they're quickly shed and gotten rid of. And the results were, under the mono conditions, E. coli produced few to no tumors. But they, in contrast, saw pronounced tumor induction in the co-colonized mice, and that this tumorigenesis required the presence of both the Bacteroides and the Colibactin genotoxin. So not only do we know the microbe responsible—
that's the E. coli toxin that does the damage, right?
Right. So not only do we know the microbes responsible for triggering the event, we know we need two. We know the toxins responsible. So, and we know that's the case because from when they do this next really cool experiment where they do an in-frame deletion of the BFT gene from the Bacteroides, and then they delete the PKS virulence island, and they saw significantly reduced tumors. So, I mean, that's
they also have survival curves.
Oh, yeah, the survival curves. Since the audience doesn't have access to the beautiful survivor curves, I didn't want to torture their minds. That they can probably see this paper. Science is probably in many of the libraries that folks have access to, and so they can probably see the survival curves. And they're the easiest things to interpret because it's all or none. I mean, and it's significant not only to a p-value of 0.05, it's significant out to p.0001. Talk about a smoking gun. So no discussion of cancer would be complete without, introducing the immune system. So they introduce us to IL-17, which is an interleukin that's a pro-inflammatory cytokine. So inflammation is bad. We know it exasperates cancer. IL-17 is produced after it's been stimulated by IL-23, and that's the extent of that aspect of the immunology. But they have this second model, the one involving the APC min delta 716. And they blame the enterotoxic B frag because the B frag toxin was necessary for the synergistic tumor induction under the co-colonization conditions. And they point you back to the figure where they actually illustrate that elegantly. So now they then add the further spin of putting in this pro-inflammatory cytokine. And as they reported, when they measured IL-17 expression by quantitative PCR, they saw no significant difference. So PCR is only measuring gene expression. So they're looking for the expression level of IL-17 mRNA, and they saw no significant difference in the overall mucosal IL-17 mRNA levels between a 15-week ET, the enterotoxic B frag monocolonized, and the co-colonized B frag with the E. coli. But co-colonization of IL-17 deficient mice ablated tumorigenesis. So IL-17 is somehow involved, but they didn't see any changes in the amount of messenger RNA. And that's probably a consequence of the redundancy of our immune system being able to get to different spots by different routes. So the immune story is not as nailed down as yet, but it's still a component of what's actually going on. They then take us down further the immune system by looking at flow cytometry, asking the question. They tease this apart by looking for early colonic mucosal IL-17 production by using a germ-free system that were mono or co-colonized, and asked what's going on by looking at the innate and adapted lymphocyte subsets. And what they learned is that the germ-free mice co-colonized with these two bacteria displayed a trend towards an increase in total mucosal IL-17 producing cells when compared to the monocolonized forms. So driven by both the adapter, which is the T17 helper system, and the innate, which is particularly the GD-T17 cells. And so while necessary for tumorigenesis, IL-17 alone appears insufficient to explain this synergistic tumorigenesis seen in these co-colonized mice, since robust IL-17 induction in the animals receiving only the enterotoxic B frag genes were only observed to have this meager colon tumorigenesis. And
is there a way you could sum up this part?
The immune system is involved.
Part in take-home lesson. Is there a take-home lesson here?
The immune system is involved in the formation of the tumors. Inflammation is important, but they haven't teased apart the complete story yet. And
that's clear.
And as they begin to take us through this, they then begin to talk about mucus depth and who's giving access to the endothelial layer, or the epithelial layer of the colon. And they're asking the question, is it E. coli colonization alone? Does it have an impact on mucin depth, or do you need somebody else? And here's where they bring in the Akkermansia, which is the known human colonic mucin degrading bacteria. So what they concluded from this set of experiments is the order of addition for mucus degradation by the enterotoxigenic B frag promoted this enhanced E. coli colonization in this niche. So if you will, the Bacteroides is the founder and E. coli comes in, and you almost can take it back to the classic AB type toxins we talk about in bacteria, where one of them facilitates the binding of the toxin to the receptor, and then there's the active site of, if you will, the toxin. And so the B-frag is the equivalent of binding to the colonic wall, and then it facilitates E. coli coming in, supplying its genotoxin, or its colibactin, the DNA damaging toxin released by E. coli to the colonic epithelial layer.
If you go back to the early discoveries of pathogens in the 19th century, where you usually found, in the majority of cases, a single bug. Here you got two bugs leading to the pathology, and that is, there may be there are quite a few other cases like it in the literature, but this one is really very, very, very marked. It's a strong case for that.
Yeah, and so here's where they leave us. They begin to put it all together for us. Taken together, all of their data collectively suggests that co-colonization, first with the enterotoxic B-frag, and then this pathogenicity island containing E. coli that makes this genotoxin colibactin, promotes enhanced carcinogenic through two distinct but complementary steps. The first, mucus degradation, enabling increased E. coli adherence. But if you think about what E. coli normally does well as a pathogen, it sticks. It adheres. I mean, that's effectively how it causes UTIs. It adheres. And so the mucus degrading enables E. coli with this colibactin to adhere, inducing increased colonic epithelial cell damage by colibactin. And then we have the immune system. IL-17 is induced, promoted, if you will, primarily by the enterotoxigenic B-frag's toxin, with early augmentation by the colibactin E. coli containing strain. And so they propose that these mechanisms yield this cooperative tumor induction. And as Elio said, this is a heretical concept. It sort of violates Koch's postulates because you need to recover two organisms in order to get disease. And it's absolutely a fascinating story of how they take it through. And I'm going to do what Michelle loves to do. And
before you do that, let me just interject.
Sure.
So I asked you earlier who has these bacteria. So they answer that here. I forgot. ETBF and PKS E. coli commonly colonize young children worldwide. So a lot of young kids have it. And so the idea is you have these at a young age, it could contribute to colorectal cancer, which you get later in life. You've got these all throughout your life. Now, in this study, they get the bacteria from patients with familial adenomatous polyposis, right? But they say this could even be an issue with people who develop sporadic colorectal cancer, because in most of those people, you have loss of this APC gene, and they showed in this paper that mice lacking with a mutation in APC, they will get tumors caused by these two bacteria. So the possibility is that these two bacteria are involved in a vast majority of colon cancers, which would mean we should screen for it. We should do a clinical trial of people with all kinds of colorectal cancers and see if they have these two particular bacteria. And if so, it would be a great screen early in life to look at your likelihood to have colon cancer.
It occurs to me that if you wanted to put this to use and prevent this kind of cancer in people who are susceptible to it by virtue of having the right bacteria and the right immune system, that you might want to think here of using bacteriophages.
Exactly.
Antibiotics are going to clean up the gut.
Yeah.
And that's not.
No, it's right. Use phage specific for these bacteria. Great idea.
Or even better, you can use CRISPR-laden phage to come in and trim out the toxin out of the genome.
So phages is the specific agent instead of antibiotics. I mean,
absolutely, I agree with that.
This paper may lead to something actually useful. It seems possible to me that knowing all this stuff, you can do clinical trials in which you try to get rid of the specific bugs.
Yep, I agree. Very exciting.
So, as Michelle,
great paper.
as Michelle left me with the first author's bio, she wanted me to make mention that Dr. De Gea was born in the Bay Area of California. She's always loved science, had a microscope even as a kid, where she studied lots of insects. She got her Bachelor of Science degree at UC Davis, and Med Micro was her major. She then went to earn her PhD at the Johns Hopkins University with Cindy Sears, where she switched to her lab in her third year seeking this translational research linkage. And I think she really got it. This science paper is only but one chapter of her thesis. She also has other papers in PNAS, cell host microbe, mucosal immunology. And as I mentioned earlier, she pioneered the development of technology to detect biofilms on tissue sections that they have patented. And as to the most exciting day in the lab, it's the day they saw the dramatic results from their first pilot with the mouse cultivation experiment, where these two organisms had to be present. And I think we can all appreciate that. And then Michelle writes in her note, she was especially proud and excited over the second figure, where the significance factor was, P.001 that I pointed out in the narrative. The other thing that the Christine points out to Michelle in her conversation is she credits Dr. Sears with making connections with scientists with different expertises. And as you look at the author list of this paper, you can see this is truly team science. There's expertise from immunologists, there's expertise from cell biologists, surgeons are involved to get the samples. So this is really a collaborative translational event. The other thing that Christine really wanted to highlight is to credit Payam Fateh, the second author, because this individual was talented and a devoted lab tech who would meet Christine at 5 a.m. every morning to begin their mouse dissections. This individual is now in a PhD program at Vanderbilt, pursuing his interest in microbiology. And when Michelle asked her the famous question, what advice would you give to other students and trainees? Christine remarked, have courage and confidence to follow your own interests. And at times you have to follow your gut instincts about what observations are important and interesting. And she was especially most grateful for her mentor, Dr. Sears, who gave her the freedom to pursue the side projects. And this, folks, was a side project. So, I mean, this is incredible.
This happens so often, doesn't it?
Yep.
Yeah.
Sure.
Right now, Christine has gone on. She presently is working for our Food and Drug Administration at the Center for Drug Evaluation and Research, and she's in the Division of Microbiology there. And she's responsible for evaluating processes for manufacturing biological therapeutics to ensure low risk of contamination with microbes or products like LPS. She's also the proud mother of two children, ages 2 and 4, that were born as she was preparing to defend her thesis and during her first year of postdoc with Dr. Sears. So again, this was a science paper that I stumbled into on an airplane, and it turned out to be one of the most remarkable reads I had on that particular trip. So
really good job. Thank you, Christine. Really, congratulations. Keep up the good work. Let me read a couple of emails from our faithful listeners. Harry writes, Dear TWiMers, I hope my entry for the book is not too late. I'm sorry, Harry. It is. I enjoyed Dixon on TWiM, and I look forward to more cross-pollination of the Twix hosts for their insights. I've been listening to the Microbe TV podcast since I took microbiology as an undergraduate in the SUNY system. The shows are so entertaining that I have continued to tune in as I completed a master's in microbiology and immunology at Colorado State University. I did some work with the school's biosafety department and have been interested in such matters ever since. I now work as a quality technician in medical devices where I get to combine my micro education with engineering. Thank you all for your dedication to science communication. Amir writes, Hey, my name is Amir and I'm a Master's of Science student of food science in the Hebrew University in Israel. I love your show. I listen while commuting to the lab and where I'm trying to develop a quick spectroscopy-based method to detect bacteria in drinking water. I'd really like to get this book about disinfection because I had a discussion with my PI about how exactly does chlorine, ethanol, and boiling kill bacteria? Do these treatments break the cells, break proteins and other molecules? And most importantly, do they make the bacteria lose their optical fingerprints? I would like to hear your opinions on this question, by the way. Thanks a lot for the podcast. Michael, how does chlorine, ethanol, and water and boiling kill bacteria?
Each one is separate. Ethanol and boiling are a little bit different, but what you're effectively doing is changing the protein structure. We've all seen that television commercial. This is where they crack an egg into a frying pan, pointing out that this is your brain on drugs, and the proteins congeal. Well, that is effectively how heat kills. It's of course coagulating and changing the tertiary structure.
Wait, chlorine, I think alcohol works first by dissolving the cell membrane.
Absolutely.
I mean, it just breaks it up.
And
so that's the first thing it does. And when that happens, after that, it initiates the proteins. But the cells are dead the moment the alcohol hits them because the membrane is gone.
That's right.
That's how the disinfectants work that I used all over the place. It was 66% alcohol.
Yes. And the other aspect is it will change the way they look because it depends on what you're actually looking at in your mass spec. And it's time of flight and whatever else is going on. So, but you just have to have a good database of, you can boil for a prescribed period of time. You can add, as Elio said, the 66% ethanol and similarly the hypochlorite, which will effectively add oxygen to everything. And you can see what it will look like.
Do they destroy the optical footprints of the bacteria?
Well, if he's using mass spec, he's
I once
probably
wrote a paper on that. A very small amount of hypochlorite will break up bacteria like there's no tomorrow. 10 to minus 5 molar is just enough to just break them up.
Yeah.
Interesting. All right. One more from Augie. Elio will like this one. Dear TWiM team, at last I decided to drop an email to the cast of my favorite podcast. I started listening to TWiM following a friend's recommendation sometime last year, and I chewed through the 140-some episodes in just a few months. This podcast should be a mandatory material for graduate students, as they could learn not only the science part, but also the logic of analysis, succinct description of complex topics, and the overall importance of communication in science. I wish you could produce an episode every week, but I understand the reality of a busy PI. At least I can rely on the Immune podcast, also excellent, and an occasional TWiV while waiting for the next TWiM. On a side note, I would like to mention that I was lucky to see Elio's Tales from a Blog talk this past December in Boston. How about that, Elio?
Well, that's available.
It was recorded? He heard you talk.
I'm sorry?
Augie heard you talk.
He's at BU.
Tufts. He's at
Tufts.
Yeah.
They were at Tufts.
Anyway, he was lucky to hear your talk, Tales from a Blog. Needless to say, it was fascinating, even though I was familiar with most of the stories, as I read In the Company of Microbes. I was hoping you could sign the book for me, but unfortunately I forgot to bring it. By the way, in my lab, we have an Eppendorf rack used by Elio back in the day. I'm keeping it as a lucky charm. How about that? He's got your old Eppendorf rack.
I can't guarantee that, but
and
give it a try. Can't hurt.
Augie has two papers that he suggests. Microbiome paper going beyond your typical sequencing and show a cause-consequence and not only correlation. Elio should be pleased with this. And another paper, a fascinating tale of emergence of Clostridium difficile with some help from a dietary supplement. I hope the whole TWiM team stays together for many years to come. Augie is a postdoc in molecular biology and microbiology at Tufts University, which I guess is where you were, Elio, right?
That's right.
Very good. Okay, thanks for your letters. We have more, but we'll save them for next time. This has been TWiM 171, which you can find at asm.org/TWiM. And if you have a...
Boy, how time flies, huh?
You bet.
Yeah.
If you have a phone or a tablet and you listen to podcasts, just subscribe on your favorite player. Please subscribe. You get every episode. And if you love us, consider supporting us financially. You can go to microbe.tv/contribute. A couple of different ways you can do that. And finally, if you have questions or comments, Send them to TWiM@microbe.tv. We don't have a book contest today. We will next time. Have plenty of books to give away. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent. Thanks, Elio.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM and Ray Ortega for his technical help, and music by Ronald Jenkins, ronaldjenkins.com. Thanks for listening, everyone. See you next time on This Week in Microbiology.